Good morning, everybody. Hi, how are you? Come on in. You heard earlier who I am, and I'm still trying to figure out where to put this thing down. It doesn't quite fit in a pocket, so I'm going to try this. There. Okay. Hi, my name is JP Clancy, and a recent addition to the CF Foundation, and I am talking to you today about tackling nonsense and rare mutations. This is really informal. In fact, I'd be walking around, but I'm on a camera, so I can't. I know several of you, but just to give you my background, I got connected to CF back in 1988. I was a medical student, and I was just finishing my third year, and I couldn't decide between pediatrics, internal medicine, or an OBGYN. And so I had one of my best friends was an MD PhD student, which means they get both degrees while they're sort of doing this journey of medical school. And he was working in a research lab that was studying CF, and he basically just said, well, do you want to try getting a job here? So I got an American Heart Association fellowship for the grand total of $1,000 a month, okay? It was awesome. It was like the most money I'd ever made in a month in my life. But I worked in that lab for a year, and that lab happened to be of a guy named Mike Welsh at the University of Iowa. For those who don't know Mike, he is a real pioneer in CF research. He is dare say, and this hopefully doesn't come out wrong, the father of the CF pig. But anyway, he has been a leading mind in CF for 40 years, and I had the opportunity to work with him sort of very happenstance during medical school. And so that year, that started as 1988, became 1989. Lo and behold, we were trying to figure out this chloride channel, what it is, how it works. Is it this certain type of channel that causes CF? No, it's that kind of channel. There was all this debate going on, and then somebody found a gene that causes CF, and all of a sudden, the way we approach CF just flipped overnight, and the way we try to understand the disease and how do we approach it was complete. It was like sort of like pulling back the covers or turning on the light or whatever analogy you'd like to use. And so, so I got into it for the science, and then after that, I decided pediatrics and went to the University of Virginia. Really didn't see many CF patients because they were cared for at a sort of a slightly different facility, a rehab facility, which is a little unusual. Went to UAB, the University of Alabama at Birmingham, because I really like this guy, Eric Sorscher, who was also a big CF researcher. And that's honestly where I started getting the most exposure to CF patients. And I was hooked quickly and forever. And so worked there for a number of years, went up to Cincinnati Children's in 2011 to sort of help grow their research program around CF. It's a great institution, but its CF presence was not where it could be, and so I had that opportunity. And then, starting this year, joined at the CF Foundation. So anyway, that's the 1 minute JP Clancy background, and I am an awesome dancer, I will say, but and my socks are also awesome. So my current title is Senior Director of Clinical Research. I work quite a bit. I sort of have three focus areas, if that makes sense. One, TDN-related projects. I'm sort of assuming that role over time. Two, I help oversee the Clinical Research Awards programs, which is really interfacing a lot with investigators at different institutions. And three, physician and physician science training. That's really my areas of interest. So I work quite a bit with other groups, no question, but those are sort of my lead things that I do, and they're all very close to my heart. So I wanted to give just, again, a brief summary of, about the landscape of CFTR therapies, literally a slide, so it isn't long. Obviously, we're going to be talking after that very meaningful and powerful presentation. I was telling Cam as I walked in, this would seem like the perfect moment for the rest of the crowd to jump on stage, and then I foresaw a catastrophe of a collapsing stage. So I'm glad my impulse was controlled on that one. But anyway, we'll talk a bit about it. And I'll focus in on sort of three strategies that we're taking. We touched upon theratyping, modulators that really are approaching nonsense-mediated CF, and what are these DNA and RNA therapies. And we've had a hard time sort of putting them under one umbrella, but hopefully it'll make sense once we're done. Mike showed a different picture of CFTR. This is the much more new and improved picture of CFTR. So what is CFTR? It is a protein. It's a gene. A gene is sort of the blueprint within the cell that gives the instructions to make these things called proteins. And proteins go and do functions in cells. And the CFTR function is to help move salt and water. And specifically, it helps chloride and bicarbonate move, but it also helps regulate sodium movement. And without getting into the gory details, that sort of helps impact how water moves, and it also impacts how mucus is formed. And we believe when CFTR is missing, well, we know when CFTR is missing, all of those things are thrown off, and they lead to the symptoms that we know as CF. Lessons learned from CFTR modulators. You know what? This seems so obvious, but if you step back to 2011, we didn't really know that if you fix CFTR, that would make any difference for people. It seems like such an obvious question, but no one really had ever done it before. There have been tries with gene therapy that, early days that were not successful. But what we learned from this is just the key data from the ivacaftor in G551D study. Small group, about 4% of CF patients. And what we saw was unprecedented improvements across everything, essentially. Looking here, we look at lung function. The guys in blue are the treated patients, and the people in gray are the not treated patients. And you see it pops up and stays there. The thing over to the right is pulmonary exacerbations, and basically we've cut that in half, or the drug does. CFQR, those are symptoms, respiratory symptoms. Those improve. That's what we see, that increase there. And then weight. And if you keep looking, if you look through subsequent studies, we found that mucus clearance actually gets better. There's some evidence that actually in younger patients, the pancreas gets better. There may be some evidence that CF-related diabetes is improved. Maybe we're starting to change infection in people who've had them. So point of all that is CFTR makes sense. And that's why we spend so much time trying to fix CFTR, replace it, edit it, take your pick. CFTR is a really logical target. Oops. There we go. And why does this fixing CFTR matter? Well, in the short term, I just showed you, over a year you can show this. But this is just a projection based on that data, which honestly is probably, think of this kind of, I think, is the worst case scenario. The red is sort of a person who, for example, has a G551D mutation, using kind of available therapies now, what we would sort of project that lung function does over time. And we see that this gradually declines over time. It's obviously not a straight line. It's got ups and downs. But over time, sometime in the mid-40s, we would expect that on average patients would have pretty advanced lung disease. If you add ivacaftor to the equation, that slope changes completely. And in fact, what we think is if you add it before there's damage, it may completely stop. But if you even start with lung disease, you can actually cut that loss of lung function in time by at least a half, maybe more. And so what that leads you to is that patients who are living into their 70s and 80s with CF and not dying of CF. And so that's why we came to the conclusion that this is such an important target and why we should put so much effort into it. So the pie is here, and I wanted to talk, where we're going to be going for the rest of this talk is really focusing on that roughly 7%. This are people who have two mutations without protein. They maybe have a rare mutation, and the second one's a non-F508 that doesn't sort of fall into the Kalydeco or Symdeco group. Might be other mutations that just aren't well characterized, and there are people out there who still don't have known CFTR mutations. While the 90% include those with delta F in 2 copies, delta F in a minimal function that, as defined by the triple combo studies, and a partial function or residual function is another word, and patients who have gating mutations, and these are the patients who are candidates for the medications now, and we believe in the not too distant future. And talking a little bit about the rare mutations, it's kind of, and I hate to segregate everything, but it does help me think a little bit about how you go after these different types of mutations. As we know, you can debate the exact number, but somewhere around 1,700 known mutations in the CFTR gene. What that basically means is that there's about up to 1,700 ways in which the gene can be broken, and they're not all the same. People put them into different classification groups, but I think an important point is that there are about 1,000 of those mutations in which 5 or less people actually have them. And I think that makes it quite clear that those mutations clearly are rare, and they're very hard to study in a clinical trial as we think about normal clinical trials. If we break that 1,700 down into sort of groups that make CFTR protein and those that don't make CFTR protein, and what that means is that the type of mutation, when it makes protein, it might just make a single little change in the protein structure that allows you to have a protein available. It's just whether it's actually able to be help it to get to the cell membrane or help it to be opened. There's a lot of different ways, but at least there's protein, something to work with. And that all these drugs that are currently in the pipe, or excuse me, that are in the clinic, are drugs that actually work on the proteins. The other roughly half are mutations that make very little, if any, protein. That includes nonsense. It also includes some other ones that you may hear about. You know, there's splice variants. There's things called deletions, frame shifts, where there's literally everything's off by about one instruction letter, and it kind of throws off everything that follows it. And unfortunately, most of these won't respond to the modulators. And in fact, as many as about 5% of patients only have these mutations. So this is one way to start thinking about how do we approach these two different, or not different, but these things that probably wouldn't be logically approached the exact same way. So strategy one, again, we touched on this concept of theratyping, and basically that's kind of better understanding a CF mutation based on how it responds to a drug. And we use modulators, we use other conditions too, honestly, to better understand the nature of these mutations. But most of these types of mutations fall into a category that are called missense, and missense basically means sort of there's one little change. You know, most of the protein, in fact, 479 out of the 1,479 of the amino acids are the same, and one may be off. And that change, though, is sufficient to get in the way of how that protein works. But since they make protein, it's actually got something that's testable. You can actually take these modulators and other conditions and actually have something to actually try to test. And so to do that, the first studies of this have really tried to standardize the way we do those studies. And you may hear these things called FRT cells, and you know, what the heck is an FRT? It's a Fisher rat thyroid cell. Why we use Fisher rat thyroid cells is kind of beyond me. It kind of is a cell line that's been around for probably 30 years. It grows easy. It kind of makes a sheet of cells, and so it's an easy one to put genes into if you want to understand that gene. So the point of that, though, is it's got to be kind of standardized. You got to be able to do this in a way such that it really gives you kind of reliable results, meaning you're going to get results the same time over and over if you do the testing. And so this has been used for ivacaftor to extend the label, and Mike Boyle was talking about this earlier. This is from, this is one of these very sort of difficult to read, but hopefully conceptually makes sense types of slides. What we're looking at is each one of these bars is a different CF-causing mutation that's called a missense, which has been put into these cells, and these very standardized cells. And the dotted line is about 10%. That's the 10% mark. And what 10% means is 10% of wild-type CFTR function. The black is sort of activating CFTR the normal way, and the white is the added boost you get out of a drug like ivacaftor. And what you can see is that for these mutations, first of all, they have a whole variety of different activities baseline, but there's a group of them that seem to go do a whole lot better when you add ivacaftor, and those are the guys that I've circled in red. And what the FDA has been willing to do is to look at those mutations, and they've sort of decided that if it's above 10% of what we would consider normal, then that would be sufficient to allow us to extend the label of a given drug. And so through that process, the FDA has opened the label of ivacaftor, for example, to multiple additional mutations. I think it's 38 additional mutations, really based on this sort of laboratory testing. And as I alluded to before, this is really best served. This is not going to answer kind of safety data and sort of that sort of thing. These are really probably most logical for drugs that find its way to the market, and then we can start determining whether they're effective in additional populations. Another slide, I'm sorry, I'm going to warn you. This is the slide that he showed before, but I'm going to make it awesome, okay? So that was looking at about 50 mutations. The CFF lab, in conjunction with two other labs, have actually taken this a step further. They've looked at every one of those 658 mutations that make protein. And they've tested not just the responsiveness to ivacaftor, but also ivacaftor and lumacaftor, ivacaftor, or Cambi. And this has been done in 3 independent labs, and they crisscross their data, and they give you this. What are we looking at? Well, what we're looking at here is this is kind of a measure of CFTR activity. And you got to remember these are a cell line, so it's not exactly the same as looking at cells derived from a patient. But the black line is sort of the background activity that we can get. The yellow is sort of the added bump out of ivacaftor. The red is the added bump out of lumacaftor. And the green, oops, here we go. There we go. The green is the added bump you get out of Orkambi. And the point of it is, no, it doesn't work for everyone, but it works for quite a few. You can see some of these guys popping up here. These are the guys that have very little function. Some of these have a little bit of background function. And this here is the 10% line again. And the point is the addition of Orkambi starts to push a lot of these guys up into the area where we think they might be effective. And so this strategy, I mean, first, this data has been shared with Vertex, and Vertex is also doing this. Ultimately, it's up to Vertex to go and bring this to the FDA because it's their drug. But we've been able to sort of give them a nudge, say, by the way, if you look, you actually can see some very good evidence. And what we don't know is triple. Does triple even do anything more on top of this for some of these other missense mutations? So I hope I can just give you the concept here that for patients who have rare mutations that are missense, that there are ways, and they're very rare and not able to do clinical trials, there are ways to sort of get at whether there's any evidence that they might respond, and we're hopeful that's going to lead to some label expansion. So I wanted to then spend the rest of the time really talking about approaching the last 7% and really focusing on things around nonsense mutations, approaches to bypass CFTR, which we won't spend as much time on. I sort of hit on some of this earlier, treating sort of some of the symptoms and some of the other sort of non-CFTR targets, and then the concept of sort of replacing CFTR and where we are. And again, as I'm talking, please, stop me if I'm not explaining something well. I'll talk first about nonsense mutations. There's been a lot of work doing small molecule screening. What does that mean? That basically is the starting point for making modulators. Modulators are basically, they follow in sort of a type of drug called a small molecule. They sort of have a certain size and range, and these are typically drugs or medicines that you can take by mouth, sometimes IV, but usually by mouth. And there's a lot of screening going on to find what those kind of compounds are, which then ultimately get turned into, they get sort of modified until they become something that's a drug, something you could take, absorb, goes throughout your body, that sort of thing. In addition to that, some other approaches which are a little more cutting edge. On the other side, we'll talk a little bit about mRNA, and I'll walk through what exactly that is in just a minute, and also DNA approaches. When most of these are really focused on, these are sort of, one of the DNA is what we consider gene therapy. RNA is sort of a new concept that really wasn't available even 5 years ago, that's really been gaining speed. And then to also know that, we know CFTR is a regulator of chloride transport and sodium channels. We know that we believe that if we can kind of restore that regulation, even if it's not through CFTR, we might actually be able to demonstrate some benefits. If we could slow down the sodium transport, we could activate additional or different chloride channels to sort of substitute for CFTR. We may actually be able to sort of have airways and epithelia that are actually moving salt the way they should, even when CFTR is missing. So I just wanted to just kind of highlight some of the investments that are ongoing, and I think Mike's might even be more up to date. I think this was about January, and so things move kind of quick. And I won't go into great details, but just to highlight, this is nonsense drug discovery, RNA, gene therapy, gene editing, a variety of other sort of approaches that sort of don't fall easily into those categories. And you can see here in 2018 and so the projected budget commitments that the CF Foundation has, and it's somewhere in the $72 million range that really is being focused on sort of developing drugs and therapies for this group of patients who currently are not benefiting from CFTR modulators. So this is my really bad cartoon, and I apologize in advance, but this is trying to understand or explain what's going on with nonsense-mediated CF. There's different ways people describe this. There's stop mutations, there's premature termination codons, also known as PTCs, but nonsense basically means that there is a stop sign that's in the gene that doesn't belong. And that stop sign, though, is sort of in the right place, such that everything before it and after it's okay. It's just right at that spot. There's been what's called a base substitution that tells the gene to stop making the protein that it's supposed to be. And so what we have here is, this is a ribosome. Isn't that great? And the ribosome is, it's amazing what, who needs a graphics department at the CF Foundation when you got this going on? So you have a ribosome here, and its job is to read the RNA and to make a protein. And so here we go. This is pretty high tech. You ready? Wow. There you go. So it hits the stop sign. Thank you. But there's kind of two problems I wanted to highlight. One is it hits the stop sign, and two, when you have a stop mutation, it makes that mRNA that's underneath unstable for reasons that we're starting to understand and somewhat above my pay grade to understand. But the point is, there's two problems here. You got to get past the stop sign, and you have to have enough mRNA to actually be able to read through. So what that creates is sort of two targets we need to be thinking about as we tackle this problem. Suppressing is the word they use for or causing read-through of the stop mutation, and also what's called reducing nonsense-mediated decay. You may hear that word, NMD, and basically that means you're keeping the mRNA template stable in levels. And if you can accomplish those two things, we actually can make a functional protein that comes out of that. There's certainly been a lot of investment, and this is highlighting over time. Unfortunately, I think we're aware that Adelerin failed to meet sort of the goals or the endpoints that we had hoped for nonsense-mediated CF. It did receive approval for Duchenne's in certain countries, but following that has been a very significant investment by the CF Foundation in partnership, both with Southern Research Institute. They kind of made their name making HIV drugs, actually, and so they're really, really good at making drugs that have gone on to become clinically relevant. And then Icogen, which is a company which has a somewhat different approach, but has a very, very robust, meaning lots and lots of compounds that they can screen. And you can see here that there's a fair amount of screening that's already happened and is in progress, trying to identify some compounds that could become these types of drugs. So I wanted to go nerd on you, and I apologize again in advance. There's a lot of that going on. But I wanted to just show you some data that indicates you can actually develop compounds that are able to read through and produce functional protein. What I'm going to show you some data that came out of the CFF lab. And there's kind of three arrows you're going to see. The black one is the normal way your cells will activate CFTR. The blue is the added bang you get for adding ivacaftor or Kalydeco. And then the red is a blocker just to make us really confident that what we actually did was open up CFTR. And what we're looking at is three different stop mutations. On the top is Y122X, the middle is G542X, the bottom is W1282X. And what we're seeing here is in the background, nothing happens. There's very little activity. When compounds are when the cells are incubated with a certain compound, doesn't have a name yet, honestly, at 10 and 30 micromolar, what we see is that there is this activatable CFTR, and that's what these things are here. But it's not the same everywhere. What you see with a Y122X is very different than what you see by another guy, a G542X, which is also different than a W1282X. So the challenge, I think you can see here with all these different mutations, they kind of are likely to respond differently to these different types of approaches. And we understand somewhat why that is. Some of that has to do with the actual what exactly the stop sign is. There's actually 3 or 4 ways you can have a stop sign, and also what's going on around the stop sign has an influence on that. But what we've also found is that adding drugs like VX-809, which is lumacaftor, can also add benefit. When you make that protein, it actually might benefit from adding something else on top of it. And here is an example. This blue is ivacaftor's added benefit. This is the ivacaftor effect right there. And I think the point of it is that I think that these types of drugs may actually include, or these therapies may include certain drugs that are currently available. And they won't work in the absence of the protein. But if you get the protein there, you can actually get added benefit out of some of these drugs. So there are a number of programs that are ongoing, and I'm obviously happy to talk in more detail about them. I just highlight a couple or two here. One is RECODE, which is their interesting approach. We didn't talk about this, but one of the steps in turning the mRNA template into a protein is something called a tRNA. It goes and grabs something called an amino acid and brings it to the ribosome. And this approach by RECODE is actually building tRNAs which actually are able to recognize the stops and put in an amino acid in its place rather than a stop. So it's a very, very novel approach that's being taken right now, mostly in the lab at this point, but conceptually is really interesting. ELOX, have people heard a bit about that? This is a company that's actually in clinical trials. They're studying a synthetic aminoglycoside. An aminoglycoside is actually a type of antibiotic which binds to the ribosome. If you've ever been on tobramycin, you've been on an aminoglycoside. It's a member of that class. Gentamicin is another one that has been shown to have a little bit of read-through capacity in certain situations. This drug, though, is an offshoot. It doesn't kill bacteria, but it does promote some ribosome read-through. The question is it enough? That's really the big question. And so that is entering clinical trials right now, and we've been talking with them quite a bit, too. And then finally mentioned that CFTR lab is, I think this is an understatement, is actively screening drugs. But I think what they're doing, too, is also helping develop the toolkit that we're going to need for developing these kind of medications. It's probably not going to be able to follow the playbook exactly that a company like Vertex was able to do. They used FRT cells, then they went to human bronchial cells, and then they went to patients. And we're already learning that what happens in an FRT cell for nonsense-mediated CF may not be the same as what happens in the human airway cells. So we're working on developing those. I will highlight a study that's ongoing right now that's really focused on getting access to cells that are from patients who have nonsense-mediated CF. This study is called RARE, and it's been open for about a year. And it is building off our learnings that to really develop these drugs, we may need to have cells that actually are derived from patients because it has all those other sort of components in them of the nonsense-mediated decay, in addition to trying to tell the ribosome to read through a stop mutation. And so far, we've been able to enroll 50 subjects. All of them have 2 stop mutations, and we've been able to grow and bank nasal cells, bank cells that will become stem cells, and I'm going to talk to you a little bit about rectal biopsies. And I know that's just what you want to hear before lunch, but you have to embrace your things. So let's talk a little bit about patient-derived cells. Why are these important? Well, again, we believe that for nonsense-mediated CF, working with authentic cells that came from patients are probably better than working just with cells that are just laboratory-based, derived from a rat or something like that. And there's sort of 3 different types of tissue that we can work with. GI tissue, and these are what organoids or enteroids are. Airway cells, which we can brush and then grow. And then stem cells. And the stem cells are very exciting. The challenge is it takes a long time to get a cell from the blood to go back to being a cell like an airway cell or something. And it's doable, but it's challenging, I'll be honest with you. So the first I wanted to show you is this is the nasal cells that we have been able to obtain, grow, and bank for studies of nonsense-mediated CF. And what we have here, the 50 subjects, these are all their different mutations that are shown here, and this is the number of persons who have these different given mutations. And down here is the total number of vials, and what does that mean? That's about 588 million cells that are in the freezer or being grown, and once we take those out, we can actually grow those out tenfold. So the point of that is we have the beginnings of enough stuff from patients to really start being able to look at nonsense-mediated or drugs that impact nonsense-mediated CF. And this is a great starting point. However, we also know that nasal cells, they sort of have a shelf life. You can grow them for a while, but then they poop out. Oh, I see a hand. Yes. Sorry.
Uh-huh. Do they also do that for brain tissue?
Not quite yet, but that is one of our plans. We started here for the purpose of focus. What the question was, are we doing this for frame shifts, or is it limited to people with two-stop mutations, which is an excellent question, and we get it all the time. For starters, it was really to focus on building up this bank for the nonsense-mediated CF, but we intend to open it up to additional mutations. Okay. And I think the slides, are they available afterwards? I think we will, yeah. So we're happy to provide them. Sure. So, to the rectal organoids. All right. So anyway, no, why do we do this? Have most people heard of organoids? Does this ring a bell to people? Yes, no, maybe? So, sort of, kind of, a little bit? Okay, so what is this concept? It was really developed over the last 5 years. And the idea here is that we know CFTR is expressed in a lot of parts of our bodies. Turns out there's a whole lot of it in your rectum. Didn't know that. Anyway, there's a lot. And specifically in a cell type, they are called progenitor cells. And so they're a little different. They're in a part of what's called a crypt. And basically these are the cells that give rise to all the other cells that are actually lining the rectal epithelium. And these guys will grow for essentially forever. If you take care of them, they will keep going and going and going. A little ever-ready bunny. But the point is they become a source that really makes it feasible to do a really high-throughput screen on cells that came from patients. And that's why we think of these organoids as being so important. And so the organoids, what happens is you can grow them in this kind of 3D culture, and they'll make these little, they call them mini guts. They're also called, organoids are little spheres, I don't know, these little things. You can see them down here. I don't have a better word for it. But these are them looking under a microscope. They've been filled with green. And this is when you stimulate them with something that activates CFTR, if CFTR is working, they get bigger. They swell. And you can see that down below here. This is from a person who has 2 copies of F508 at starting point and then after 30 minutes and 60 minutes. And we can measure that swelling, and that swelling is just moving the water into those little organoids, and they just grow over time. And actually some very, very, very savvy and brilliant people were able to sort of capitalize that and start making that a measurement that tells us about CFTR function. And so this is just the same concept, looking at people with a variety of different mutations. And this is looking at how much they swell compared to how much their lung function improved when they started on a modulator. And so the modulator response in the organoid versus the modulator response as measured by lung function in the patient. Not a perfect line, but pretty good. There's about a 0.8, 0.9% correlation. And these are small numbers. They've been added to. But the point of this is this kind of makes us think that organoids are a very solid and logical place to be trying to test drugs before they go into the clinic and to identify them, because they seem to relate to what is measurable in patients. I'm also going to show you just a little bit, just conceptually, that it may, as I sort of alluded to before, drugs that work for nonsense-mediated CF may also need the addition of currently available modulators. They actually may synergize. They may build upon each other. And what we're looking at here is the swelling of organoids. This is the person's mutations. The point of it, this guy is the stop mutation or nonsense mutation. And this is a swelling at background. This is actually at learn. Didn't have much effect on this mutation. This is G418. It's a toxic relative of gentamicin. So it does make them swell a little bit. But if you add, or can be, you actually get a whole lot more swelling. And so the point is, it can maybe make protein, but you may actually even need to give that protein an additional oomph to really get some good function out of it. So just to kind of give you a quick, pause in regards to read-through and nonsense-mediated CF. Definitely been able to demonstrate read-through by different types of molecules in human airway cells from patients who had stop mutations. There's a good chance that we're going to need to combine things that suppress the stop mutation, which stabilize the RNA, and also maybe even help the CFTR by correction or potentiation. So you can see sort of a future where it might be a combination of medicines. Cell models are really important. And so we're spending a lot of time making sure we get those right. And then finally, this isn't the only path we should go through. This is, as you can see, it's complicated. There's a lot of moving parts to get straight. And it's probably, because of that, we think it's important to focus on additional approaches that I'll summarize in just a few minutes. So before I jump out of that, does anybody have any burning nonsense-mediated questions they'd like to throw at me? Oh, are there any up here? Oh, nothing yet. Oh, okay. Okay, sorry about that. So I'm happy to come back to them, no problem, but I just, if something was in your head, I'd be happy to tackle it right now. So the next step I'm going to go is talking about three approaches. The common theme is nucleic acid, and I'll go through what that is in just a minute. But these are therapies that actually, they actually could benefit anybody by providing a normal CFTR to people. Obviously, we're thinking it in the context of patients who aren't being reached by modulators. And we hear 7%. I think, honestly, I hate to say it, but it's probably going to be a little higher number because there's going to be people out there who don't tolerate the available modulators. It's inevitable. I mean, we all know that medications have sometimes off patients who have intolerances that just don't follow clear rules. And so I think there's going to be additional people beyond just these sort of genetically defined groups who are going to really be needing these types of therapies. So this is your cell biology class. All right, everybody ready? Okay. So, I do have one mutation. Let's see if this is one I should go. Let me tackle this after we get to this because it's a good question, but it's a little bit long. So I will come to that. This is a cartoon of a cell drawn almost as good as my cartoon of the stop mutation. And what we see here, this is the nucleus. This is where the genes are stored. And then there's this thing called ER, which we won't go through what that name is. But it is where you you basically take the DNA here, gets converted to mRNA, which then gets converted to protein. And then the protein goes up here, gets mature, and then goes up to the cell membrane to do its job where it moves salt. So drugs like potentiators work up here on CFTR that's at the cell membrane. Correctors work here on protein that's kind of stuck in the cell and can't get out. Suppressors, we believe they work down here, sort of where the mRNA is becoming a protein. And then things like gene transfer or mRNA transfer are happening here. They're sort of happening either in the nucleus or as they get into the ER. And they both have a new challenge, and that is delivery. There's been at least 18 studies of gene therapy in CF that didn't work, to tell you the truth. They didn't all go down the lungs, and these were all primarily in the 90s, early 90s. But the biggest challenge we ran into is how do you get these genes into the cells that you need to get them into. And so the good news is I think we've made some great progress and we've learned so much since that time that really makes me think the playing field is completely different. So the first I'm going to mention, which didn't exist as a concept, was RNA therapy. So RNA, again, is that sort of in between the DNA and the protein. It's sort of that the intermediary. Historically, mRNA, which again it represents the genes, just it's serving that translating function, really unstable. That's what always makes it hard to work with. It just, if you left to its own devices, it sort of breaks down on its own. We meaning the scientific community, not me, have really made some great strides in being able to stabilize RNA, which didn't exist before. And that is why I have some real enthusiasm for what's happening here. And this was one that Mike mentioned, or sort of highlighted here, that is into clinical trials. Translate Bio is doing a phase 1 clinical trial, and they're actually going to be moving they've already moved into CF patients. It is happening in phase 1, and actually what's coming up is really where the rubber is going to meet the road. Basically, what we've done so far is basically these early studies, which are sort of safety, tolerance, make sure, if you breathe it in, you don't grow a third head or something like that. But now the next step is really, can we prove that it's actually doing something? Is it hitting the target? And that's going to involve some big things. These patients who are going to be doing these studies will probably have to undergo bronchoscopy. We're going to have to look at their cells. Are their cells making CFTR, and is it working? So we're entering a new space as we go into this. But the point is, it's in phase 1, and honestly, by the end of this year, those types of studies will be in progress. And so I think this is something to be really excited about. A couple of other companies who are doing this, different strategy, but general concept is the same, are noted here, Arcturus and Acutus. And these guys, LNP stands for lipid nanoparticle. That's basically this little thing that sort of helps carry the RNA and keep it stable so it can be breathed in and hopefully get into the right cells. Important things that need to be done if we want to be able to sort of compare things from different companies or for academic labs, it would be nice to have a very established sort of animal that we could do these deliveries in. So the CF Foundation has been meeting with some of these types of labs which help provide standard animal testing. We also acknowledge that those animals that are normally available need to also probably include some CF animals, which we now have many options. And the last thing is we're working on, a big part of what I'm doing is sort of what are the early studies that we need to know that tell us that these types of approaches are really are doing what we think they are. And so I think there should be a lot of enthusiasm for this. And personally, I think mRNA is probably the most likely short-term target, I guess I would say has the best sort of short-term potential. But it's not a long-term treatment. And these are let me step back. It is a long-term treatment, but it would be a retreatment. These are not things that are one and done. You would have to be able to take these medications on a regular basis to continue to see the benefit. I'll next go into just gene therapy and gene editing, and again, these were the same. I don't know how Mike stole my slides. I just can't believe that. What is the difference between gene therapy and gene editing? Gene therapy, I think of it as you provide a normal sidekick CFTR gene. The patient still has their own gene. You've just given them an additional gene, and it may be just floating around in the cell or the nucleus, or it can actually go into the genes and chromosomes and become part of their DNA. The other is gene editing, where you basically go in. The term they sometimes use is molecular scissors. You basically fix their own gene through some really sort of amazing sort of new science that has emerged over the last 5 years. And these approaches are, they have similarities, but as you can see, they're not necessarily the same. So I'll mention a little bit about gene therapy, which I think will happen before we get gene editing, because gene editing is happening in mice, but in other animal models, and actually theoretically from a Chinese researcher. I think if you saw that yeah, exactly, a baby in China. And there's a lot of ethical things to be considered, which I think we'll get through, but we don't want to get so far ahead of our skis that and it's not ready for prime time, is my personal opinion. But speaking about gene therapy, there are a lot of approaches which are ongoing. These are just some that are highlighted. One is by a company called 4D Molecular, which is out of UC Berkeley, Tally Bio from the University of Iowa. That's my alma mater, by the way, just so you know. And also the United Kingdom has a gene therapy consortium. They all have sort of slightly different approaches going on here. One common theme, though, at least in principle, is several of them are using a type of virus that's called AAV, or adeno-associated virus. This is a virus that's well known. Most of us actually have evidence of having had it. It doesn't really seem to cause disease, but it is one of those viruses that is able to carry genes into human cells and not really create a whole lot of stir. Yes?
Just a quick question. The difference between mRNA therapies and DNA?
Yeah.
Yeah. Okay. Excuse me. The difference between the mRNA approach and the DNA approach, is the DNA theoretically a better approach because the DNA is a more stable molecule? What's the difference?
Yeah, no, fair question. Great
Which
question.
one's going to win?
Which one's going to win? Obviously don't know, but I think they're complementary. So historically, the DNA made more sense because it is a more stable thing. You can put it into, this is an example of a virus they've also been working on how to change the sort of the surface of the virus so it gets in more efficiently to cells. So in theory, historically, viruses made good sense. The challenge historically with viruses is that they can elicit an immune response, and so it may be hard to redose over time. I think we're making progress in getting around that, but it's not a simple problem to overcome. That's why I kind of think, honestly, my suspicion is we're going to see our first wins in the mRNA space. Again, that's this is me talking. My crystal ball, it's in Cincinnati. But no, I just have a suspicion that it's much less likely to evoke an immune response. And I think we probably have more flexibility about sort of dosing and so forth, which makes me feel like there's a good chance it'll be an earlier win. I think these gene therapy ones are extremely exciting. And some of them, what I like about them, they're what's called integrating viruses. You get them in the right cells.
Yeah.
They'll stay there forever. And so.
Just a comment. I love the way you dance.
Oh, stop. You're just trying to. Need my number? No, kidding. The last point. Moving right along. No, the point is there is a lot of work going on in this space. I'm showing you some viral vectors. There's also non-viral vectors, which basically are you put together a mix of lipids and DNA in certain concoctions, sort of like the mRNA, but perhaps, again, arguably a more stable approach. There's been a lot of work going on and trying to learn from the gene therapy space. The CF Foundation sponsored a summer conference last year that was really focused on gene therapy, and you can see the different areas that we talked about. We've had a subsequent one in February that's brought some of these people back and brought in some new people. You may realize it, may not. Gene therapy is actually exploding outside of CF right now. There's over 300 clinical trials going on right now that are gene therapies, and it's been estimated that as many as 40 may be approved over the next 2 years. A lot of these are for eye disease. Some of these are for bone marrow-related disease where you can kind of take the bone marrow out, fix it, put it back in. So not exactly the same. And I'm the first to tell you that the lung is not a simple place to get genes into. It spent quite a long time building a barrier to keep things out, and so it does take work. However, I think some of these strategies that I've been showing you are light years different than where we were even 5 or 10 years ago. So gene editing, this was science fiction just 10 years ago. This again is the idea where you actually are able to fix a person's gene, and conceptually wonderful, logistically complicated as you think about it. How do you have to use some special sort of, in addition to sort of bringing along the replacement piece that you want, you have to put in some other parts that are the actual, what they call molecular scissors, that get in there and put it in the right place and take it out. But it's doable, and it's being done in cells, and it's now being done in animals, and obviously in China there's reports of even doing in a person. There's different strategies. There's something called CRISPR, which you may have heard of, which is being taken by some companies. Sangamo, which has actually had some other successes in other diseases, is using a different approach. It's called zinc fingers. It's just a different way to get the target gene or the replacement piece to the area it needs to go. And then something called PNA, which are these protein nucleic acid approaches. Really exciting stuff that's coming along in that area again, and this has been out of Yale mostly, but was reported last year at the North American CF conference, showing that that actually giving this approach to some mice with CF was actually able to correct their GI problem, which is really why mice die of CF. So it's a really exciting concept that's moving forward, and as highlighted in addition to gene therapy workshops, we've also had gene editing workshops, and really laying the framework for where we think we need to be in maybe 15 and 20 years. What's the work that needs to happen now so that we can offer this? And this is kind of when I think about cure, this is what cure represents. Being able to fix their gene in such a way that they no longer have CF, and that's a wonderful, but it's a wonderful goal, and it's going to take time. So just to summarize these things, and happy to take any and all questions, and if I don't know them, I'll do something else. But just first of all, I think we've made great advancements in our science of understanding rare mutations. We have really ramped up the funding to try to tackle some of these difficult to treat problems and bring new technologies into the CF space. And I think we've really made some great strides in understanding and gaining experience with these different approaches, including mRNA gene replacement editing and also stem cells, which I didn't go into in great detail. But happy to take any questions, so please, fire away.
Thanks very much for a great presentation.
Sure.
You haven't talked much about alternative channels.
Mm-hmm. Mm-hmm.
It seems like maybe that's just been left out because it's
stuck
off
to
target.
Yeah.
But I'd like to hear a bit about the outlook for ENaC and TMM channels and also amphotericin B.
Yeah. Interesting.
I'd like to hear about the new discoveries.
Yeah, I'd be happy to talk. No, it's a very provocative concept here. So first thing, one of the things I talked about or I didn't really talk, I sort of showed, was some of the hydrator therapies that was part of under mucociliary clearance. Many of those approaches involve sort of trying to regulate alternate channels, basically. And the two ones we think most about, one is called ENaC, or epithelial sodium channel, and that's the channel that sort of regulates sodium being absorbed, and we know that sort of contributes to making the mucus thick and dehydrated. And the other ones are called alternative chloride channels, and those are sort of channels that are also in the cell membrane that maybe could substitute for CFTR. And so there are at least 3 ENaC blockers which are in phase 2 clinical trials right now. So far, other ones have not succeeded on their own. One by Perion is one example. Most people think that the failure probably represented it was more about the sort of the dosing and pharmacokinetics, as opposed to, in other words, it wasn't clear that they were actually getting to the target, meaning that they actually blocked ENaC appreciably and therefore allowed it to do what we would think it would do. And so the current studies are really focusing on, in addition to showing that they're safe and tolerable, is also really trying to prove that they actually have hit the target, so that we know for sure if ENaC really makes sense. There's also a couple of companies that are in the process of developing activators of alternative chloride channels. There's one that's called anoctamin, just to give it a name, or the calcium activated chloride channel. There's a company that's approached us about that, and we're talking with them about what makes sense and what type of partnership. But they're not as far along. The amphotericin question. So does anybody know what amphotericin is? Nope. It's an antifungal antibiotic. And basically, it's been around a long time. It is a drug which is able to. It basically puts holes in fungus such that they blow up. they can't regulate their water and salt movement. What's been found is that certain doses of this well-known antibiotic, if you put them on airway cells in the right way, in the right dose, they actually help improve the bicarbonate transport. And we talk a lot about CFTR as a chloride channel, and it's definitely that, but it also has this important role in moving bicarbonate. Bicarbonate, which is, we think, really important in how mucus forms. Mucus has to unfold the right way, and having bicarb around is really important for that unfolding to be correct. And if it doesn't happen, it ends up sort of a tangled mess. And so what this study was, which was in Nature, which is a really, really, really good journal, and included Mike Welsh, my old mentor, they looked at amphotericin. The group was out of Illinois, Champaign, I think, and they were basically showing that amphotericin, the right dose in the right circumstances, was able to normalize this bicarbonate transport, and then also improve the mucus and also improve the killing of bacteria by the cells. And so, very provocative. We've really just started to wrap our heads around what this means. But what's exciting about it, if we can sort of get some validation, this really does what it looks like it does. It's a pretty simple move to get it into a clinical trial theoretically. I mean, this is not a standard inhaled antibiotic, but it is used in an inhaled fashion in certain conditions. And there's a liposomal form, which basically is a form that is really geared towards, might be more amenable to be used as an inhaled antibiotic, or an inhaled drug. So it's an inhaled antibiotic, but maybe has actually these unexpected benefits. So, great question. And somebody's been paying attention to his thing here. I am going to try to answer one here. Our son has the W1282X nonsense gene. It's from Ashkenazi Jewish background in general. Do Jews or Western Europeans, Sephardic, carry a mutation that falls into a different, more functional class? Also, if the other gene one carries is more functional, will that one be dominant in the transport of chloride in the cell? So I think, if I'm not mistaken, the only nonsense mutation that sort of tracks with with an ethnicity is the W1282X, tends to have an Ashkenazi Jewish background somewhere. But the other nonsense mutations are not really connected specifically, but with an ethnicity. The second one, though, the part of that question is other gene one carries is more functional. Will that one be dominant? Yeah, actually, that's true. When we think about people who have, for example, a minimal function and a partial function mutation, it's the partial that sort of defines how well the CFTR works. When you have recessive disorders, like we do in CF, having a little function in one is meaningful as opposed to having a non-functional that cancels. It doesn't cancel out. It sort of, thankfully, they don't interact, if that makes sense. So having a functional would override the W1282X, if that makes sense. Yes. They can.
We're very hungry for information.
Yes.
So I really liked the phone call that Angela and Bill Sketch had set up, I think in the fall. It was kind of an update.
Yeah.
So what resource would you send us to get more information? Because we don't want to wait a year for VLC or for our annual meeting at our local chapters.
I mean, I think the bottom line is we'll set something up. I mean, I don't think we have anything on the books, but we can put it on the books. As you are well aware of Bill, who is got it. So here's my brain. There's Bill's brain. Anyway, no. Bill Scatchu is really the head of research affairs and a lot of the drug development, or even the early sort of basic science type of research, would be obviously appropriate to be in that conversation, as would Martin Mentz, who runs the CFF lab, and additional people. So why don't we circle back and we'll determine when's the right time to do that? Because I hope the message has been loud and clear. We're here for you. We are.
Just really quick, Adam, going to your question about I'm not going to even try to say the product's name, but the bicarbonate.
Yeah.
Those folks were in the office last week.
Oh, there you go.
Just so you know, I don't think JP knew that, but they were in the office last week meeting with Bill and some of his team.
Fantastic. Yeah.
Laura as well.
One more time, I'm sorry.
I just wanted to respond to the question about where to go for more information in addition to the webinar. We did last month launch a new rare and nonsense section on cff.org. So we've put some new content up on the site, and we are going to continue to add information, particularly as we announce new research collaborations and other things. So.
Oh, by the way, this is Sarah Alsbach.
Sorry, I'm Sarah Alsbach, the head of communications at the foundation. So we did announce that on Rare Disease Day, and we did use that as the hook for a lot of our content on the website to drive people there, and we're going to continue to supplement that. So it was just sort of first out of the gate to put new content up.
Thanks, Sarah. No, I appreciate the update. So I wrote back and forth, so actually things do happen when I'm not in Bethesda. So anyway, glad to hear that. Thanks, Mark. I'm just kidding. Questions? Oh, nope, you were scratching. Never mind.
More of an observation. I just wanted to say thank you because we have an amazing team, and to hear you talk, you're just so excited and could not be more excited about what's going on, and it makes us excited knowing that we have people like you on our team, and we know that we are going to beat this disease. Having said that, I'm still about 20 slides behind this slide.
Oh, yes.
So I had a question about the organoids.
Yeah, sure.
Okay, so you said you use that for testing new drugs before, I guess, it gets to actually to people for testing. Are those also what you guys are using for like gene therapy and gene editing?
Yeah, good
because
question.
Those are like the
it's a great question.
Main cells or the starter cells. I don't know what they call them.
Progenitor.
Okay.
Yeah. They are one of the cells. I think for there's kind of two sides to this. So the organoids are being used in different ways by different groups. In fact, if you look in Europe, where obviously the national health systems have an impact on the availability of current modulators, they've been used a lot in the guiding to access of therapies, honestly. I personally, I'm not sure if it's quite that advanced personally, but that's how it's being used in a lot of ways. We do think, again, another role will be around drug screening, particularly for mutations like nonsense mutations. And I think they'd be one of the one of the cell types that we would like to be able to show that nucleic acid-based therapies, and be it gene therapy, RNA therapy, are also able to restore, if that makes sense, because they are patient cells. We probably think airway cells are important too, because they're like one of those early targets, but I think this is another one of those validating cells that will be used for those types of strategies, if that makes sense. Oh, looks like I've got one of these. I think that means tie it up. So I think it'd be more like this, but anyway, that's just me, or more like this, or like this. But no, I guess the point is this has been very, I don't know, it's a great honor to be able to talk to you about where things are. And it's a big, big, big commitment of the CF Foundation. And the people who've come on board. I'm actually no longer the newbie. There's another guy named John Sheridan who just joined, who had been with the the NIH. And his focus is all around gene editing and therapies and RNA. So that's why he was brought on. The point is we're bringing on the people, and we're also committing the resources to make this happen. And so we're there till it happens. Okay. Any questions? Please feel free to come up.